Hi, my name is Janine Bijsterbosch. Thanks for listening to this talk that's part of the OHBM 2022 Symposium entitled Charting Brain Variability in Health and Disease Using Normative Models. The previous talks in this symposium have covered a lot about methodological aspects of normative modeling. And following on from that, in this talk, I'd like to tell you about an ongoing study that's happening in our lab using UK Biobank data and applying normative modeling to try and disentangle heterogeneity in depression. I'd like to start off talking a little bit more about the UK Biobank, which is the data set that we used for this study. It's a little different from a lot of other data sets in neuroimaging, and I think it offers a lot of exciting opportunities to study mental health. So this is what's called a prospective epidemiological data set. The full UK Biobank includes 500,000 people that were recruited in the UK from the National Health System. So basically they send out several million letters to anyone within the age range of 40 to 69 at the time of recruitment, and got a self-selection sample from that. They're doing neuroimaging in about 100,000 people and a second time point in at least 10,000 people. And even though this is a multi-site study, the sites were built from the ground up with the sole purpose of acquiring UK Biobank data. So it's identical scanners and identical procedures. So the side effects tend to be a bit smaller than more traditional opportunistic multi-site studies that use existing scanning infrastructure. And so this is a really interesting data set in that it is large, but also it doesn't necessarily have a specific disease focus. But there's also not a lot of inclusion or exclusion criteria. So essentially, anyone was included and participants are followed up until death. And so the idea is that the sample should reflect a good number of different disorders, both pre-existing disorders and neurodegenerative disorders that might develop over the course or after scanning. And so there's a lot of information available in terms of clinical records, ICD-10 disease labels, first occurrence data when people were first diagnosed, and then also genomic blood markers and lots of other phenotypic data. Within the context of mental health, the UK Biobank offers a really exciting opportunity. Previous neuroimaging studies of mental health have mainly been relatively small and therefore have had to have quite specific inclusion and exclusion criteria. For example, no comorbidities, not on any current medication, first episode depression, things like that. And that was absolutely the right decision if you've only got funding to scan tens or even hundreds of people. But of course, we all know that mental health in the real world is a lot more messy than that. And so I think the UK Biobank is a really exciting opportunity to study real world mental health in all of its messiness. And so that gives us an opportunity to investigate symptoms and comorbidities, to look at etiological indicators using some of the other measurements that are available in the UK Biobank. It also gives us an opportunity, given the larger sample size, to use advanced analytical approaches such as normative modeling, using a normative sample and a held out patient sample, for example. And what I'm going to focus on in this talk, although I'm going to touch on each of these elements a little bit, it gives a really exciting opportunity to disentangle heterogeneity and try and see if there's meaningful subtypes in depression based on a well-powered real-world sample. Before we get into the specific research question and methods for this project, I want to talk a little bit more about the decisions that you have to make to apply normative modeling in the UK Biobank dataset. Because the UK Biobank doesn't have a specific disease focus, there aren't any elaborate clinical interviews for diagnosis or specific labels that differentiate between patients and controls. Of course, there's a substantial amount of information, phenotypic and clinical record information available in the UK Biobank that can be used to derive such labels, but those have to be determined by the researcher. They're not simplistic labels for diagnoses are not directly available in the UK Biobank. And so I want to talk a little bit about how we defined our cohort for this study. We focused here on people over 60 to look at late life depression, and we defined a healthy cohort as a minimal score on the recent depressive symptom scale, which is a measure of depression obtained on the day of scanning that we developed and if you want to find out more information, I'd invite you to check out the 2021 paper cited here. We also looked at a variable for number of episodes of depression and make sure that our healthy cohort had 0 previous episodes. And we also looked at the measures of medication, made sure that our healthy cohort was not on any SSRI or commonly used medication for depression. The definition of the patient cohort is another question because, again, there's no standardized clinical interviews for diagnosis in the UK Biobank. I'll come back to that in a couple of slides. And then when it comes to the normative modeling, so we fit the normative modeling using the healthy cohort, which was 6,500 subjects, using covariates of age, sex, sight, and head motion. And the brain measures that we focused on here were multivariate brain measures, including resting state connectivity and amplitudes, cortical thickness, gray matter volume, and some fractional anisotropy from diffusion data as well. The goal of this project was to disentangle clinical and biological sources of heterogeneity in depression. And this project was led by my graduate student, Kayla Hannon, and I'd like to point out that she's giving an oral presentation in the session on disorders of the mature nervous system. So if you're interested in the study, I'd encourage you to listen to her oral presentation and ask any questions there as well. So the idea behind this project was that there have been previous attempts to identify subtypes in depression. One of the challenges is that people who suffer from depression differ from each other in different ways. And so one of the ways in which people with depression differ from each other is in terms of clinical heterogeneity. So these are hypothetical patients, A and B, that have exactly the same diagnosis of major depressive disorder. And I've listed a number of the symptoms down below. And it's instead of a game of find the differences, this is a game of find the similarities. And if you stared at these for a little bit, you might find that the only symptom they have in common is a relatively vague symptom of fatigue. And I mean, this is a made-up example, but this is realistically possible given the guidelines for diagnosis in the DSM. And so that means that these individuals are, of course, clinically really different in terms of their presentation. But now let's look at another example where we now compare patient A to patient C. Again, these are hypothetical patients, but now they have exactly the same diagnosis, but also exactly the same set of symptoms. So there's no remaining clinical heterogeneity. But it is possible that the symptoms, even though the symptoms are the same between these two patients, they might arrive from different mechanisms in the brain. And so I'm giving a simplistic example here. I mean, it's clear from previous work from our own lab and from other groups that the brain basis of depression is not a single region or network. It's more likely much more multivariate than that. But the idea is that even if you've accounted for clinical heterogeneity, there might still be differences in people who present clinically in the same way in terms of their biological mechanisms that lead to those symptoms. And so if this is the case, then maybe we need to think about heterogeneity and disentangling heterogeneity as a multi-step process, where we could first of all take our messy real-world data set like the UK Biobank and first of all identify clinically homogeneous groups. And then within each of the clinically homogeneous groups, look to see whether there's evidence to support that those groups break up into separable biologically homogeneous groups. And so this was the rationale for the project that I'm presenting here. And I'll talk a little bit more about how we did that. Instead of the example in the previous slide where we found two hypothetical individuals with the same kind of complex set of symptoms, in this project we isolated each individual symptom. And so we looked, for example, for individuals who had a high score on anhedonia, but a low score on these other two symptoms that I'm measuring here of low mood and somatic disturbance. And the same for individuals who had a high self-reported score for low mood, but a very low score for these other symptoms. And I should say that this is uniquely possible because of the data set that we had in the UK Biobank data. Because usually in most individuals, they will suffer from a mixture of these symptoms. And in fact, if you look at the full data set, these symptom scores are really highly correlated. So you can see out of a much larger group of individuals with depression, you can see that these groups are relatively small, but big enough to look at. So this is a really exciting opportunity to like curate these really specific and uncommon groups of subjects within the UK Biobank cohort. And the reason why we wanted to do this is because it allowed us an opportunity to identify are there different neural mechanisms associated with these different symptoms. And in addition to these symptom groups, we also looked at several other clinical aspects of the presentation of depression. So we looked at chronicity and acute severity, for example. And those can be dissociated as well. So we identified people who had a high chronicity, i.e. a high number of depressed episodes over the course of their lifetime, but a low acute state of depression, so current severity. And vice versa, we identified people who had a very high acute severity level, but a low number of episodes over the course of their lifetime. Late onset depression is where you don't have any depression before the age of 60, and then the first episode happens after the age of 60, and that has some potentially different etiological profiles. So we separated that out as a separate group. And then our heterogeneous group right at the bottom is essentially anyone that's left here that still has a high depression score but doesn't fit into any of these more unique groups. And so that will become our comparison group for further analyses. Using normative modeling, we calculated the normative deviations for all individuals in all of these different groups for 88 different multimodal imaging-derived phenotypes. And the first thing we did was essentially perform an ANOVA to test for group differences in the normative deviations across these different groups. I'm showing you example results here, and I'll show you a few more in a minute. We had multiple hypotheses going into this. One of the hypotheses was that the effect size would be the smallest in the heterogeneous group because essentially what we're doing there is averaging. If there is true clinical and or biological heterogeneity, then we're averaging across people who might have different symptom profiles and or different biological bases of their depression. And so we're anticipating that we would average out and leading to a lower average out effects leading to a lower effect size overall. And so that's something that we see in these results here. The bar right at the end is the heterogeneous group. And so you can see that that has the smallest effect size out of all of these different, compared to all of these different clinically homogeneous groups. And the other hypothesis that we had is that there might be different profiles of imaging-derived phenotypes, so different kind of brain signatures in different groups with different clinical presentation of depression. And you can see that here as well. For example, the late onset group actually having a normative deviation compared to the healthy population, whereas all of the other groups have a negative deviation. And then spanning out, we can look at these were all of the different imaging-derived phenotypes that passed a significance threshold corrected for multiple comparisons. And I won't go into the details of this, but we can see that there are that our hypotheses hold across all of these results. So in all cases, the last bar, which is the heterogeneous group, has the smallest, or at least one of the smallest effect sizes. And there are interesting different patterns for different groups in terms of the brain signatures of depression. This is ongoing work. So our second research question to ask whether there is evidence for residual biological heterogeneity, i.e., to test whether there are meaningful biological subgroups within the clinically homogeneous groups. This is actually ongoing work, but in my last few minutes, I'll touch on it briefly just to show you an example. So using clustering, we looked at and focusing on the acute severity group, we did identify two different biotypes. We did some bootstrapping to optimize the number of biotypes, and we did some cross-validation to test how stable these biotypes were. And these biotypes seemed relatively stable with an adjusted random index of 0.73. And when we're now starting to look at some follow-up analyses to validate these biotypes and see whether they are meaningful different using some measures of etiological indicators, some measures that might represent different pathways through which people might have become depressed. And so it was interesting to see within this acute severity group that there was a significant difference in simple digit substitution performance, which is a measure of cognition. And so that might be related to some hypotheses about depression in late life being a prodrome of Alzheimer's disease. But again, this is very preliminary work, and we're excited to delve deeper into this and tell you more about this in the future, hopefully. So to summarize, in this study, we aim to disentangle heterogeneity in late life depression, particularly differentiating between different sources of heterogeneity, such as clinical versus biological heterogeneity. I'm really excited about population data sets like the UK Biobank because I think it offers a really unique opportunity to take a different experimental approach, such as curating the clinically homogeneous groups, and also to make use of more advanced analytical approaches, such as normative modeling, which is really powerful to estimate individualized deviations from the norm. And these deviations can then be used in downstream analyses in different ways. Of course, I showed you some preliminary results in terms of our biological heterogeneity, but we're doing a lot of ongoing work to validate the clinical value of these subtypes. And so we're working on null distributions and using additional measures for validation. And so with that, I'd like to thank all of the collaborators that were involved in this project, my lab, and in particular, Kayla Hannon, who expertly has led and continues to lead this project as we go forward. And I'd like to invite you to attend her oral session to learn more about the details of this project and ask her a little bit more questions. I'd like to thank the funding, and most of all, I'd like to thank you for your attention.